In this segment, I'm going to be going through transcription and translation, and the depth of the information is going to be based on the amount that you need in order to solve problems about the development of body parts, the control of events in organisms, and it's something that we're going to use throughout our course in zoology. So first, a gene. Remember DNA, remember A's and T's, and C's and G's pairing up, and remember groups of 3 are coding for what will be a codon in the messenger RNA, and remember that in messenger RNA, instead of T's, there are U's. Okay, so I won't keep drawing 2 strands. Instead, what I'm going to start doing is 1 strand to represent a gene. And remember that there are coding regions of genes and regulatory regions. I'm going to talk about the regulatory regions as if they're all upstream from the promoter, the proximal binding element for the initiation factors and for RNA polymerase at the promoter. So there's a binding site for RNA polymerase, which is the protein that is actually going to produce the messenger RNA. There are proteins that bind in that area that allow RNA polymerase to bind. And then there are regulatory regions, most of which are scattered upstream, that are transcription factor binding sites. Transcription factor binding sites are sequences, short stretches of DNA on the order of 4 or 5 to maybe 10 or so bases at which particular proteins can bind. And so a particular sequence, maybe here, might have a particular transcription factor, so transcription factor A. Okay, so that's the structure of a gene that would have transcription factors binding in different locations. And the locations in which they bind and the ways they interact with the proteins that initiate transcription affect whether or not that gene is expressed. So let's imagine, here's the coding region of gene X. Remember, it has exons and introns, exons expressed. And I'm going to draw one here that has 2 exons. And when RNA polymerase moves along, it separates the 2 strands and it makes a messenger RNA that complements one of those strands. So as it moves along, we get a messenger RNA strand unrolling. Remember, A's and C's and U's and G's. And then that messenger RNA, once it's completed, the intron portion is removed. The exon portions are spliced together to make the strand of messenger RNA that is the final strand that will then be translated. Whether or not this happens depends upon the combination of transcription factors. So here's gene X again, and let's imagine that there's a site here, a transcription factor binding site, that binds transcription factor, the protein, TFA, if it's present, will bind here. And let's imagine that there's a location right here that attaches to some of the proteins that are necessary to get RNA polymerase to bind and start moving. That makes it an enhancer because RNA polymerase is moving along and we're going to get messenger RNA. And of course, it can be combinations of transcription factors. It could be that we actually need multiple ones. It could be that we need multiple transcription factors, that we actually need to have TFA, TFB, TFC. In fact, it could even be that there's some other distant one. Here's our strand going somewhere else, and there's yet another one, and that all of these together then interact with the proteins that actually, again, get RNA polymerase present and able to move and cause transcription. On the other hand, let's imagine that there's a binding site, say here, that functions as a repressor for this gene, and if something is bound there, it actually prevents TFD from binding, and that in the absence of TFD, that actually prevents all of this. In that case, then in this location, transcription factor E is a repressor because it blocks transcription factor D. You don't get RNA polymerase binding, and there's no messenger RNA produced. Okay, so that's elements of gene structure that relate to regulation. And then for translation, the messenger RNA strand, remember for gene X just has the 2 exons, is translocated out of the nucleus. It binds to a ribosome that has a large and a small subunit, moves through that ribosome, and transfer RNAs come in, which have sequences that match particular anticodons, and each one has a specific amino acid that is related to that anticodon. So each anticodon matches a specific amino acid. There would be 3 transfer RNAs in here. Each one has its own amino acid, and they would be joined into a growing protein chain in this process. So we have transfer RNAs coming in, but only if they match their anticodon to the codon and the growing amino acid chain. The end result of this would be protein X. And protein X might be a transcription factor, might be an enzyme, or it might be a structural protein, such as actin and tubulin. Is there only one combination of transcription factors that can cause expression of a particular gene? The answer is no. There can be many dozens or hundreds of transcription factor binding sites for a particular gene, and different combinations might cause transcription or repression. So in one cell, it might be the case that TFB, TFD, and TFE cause transcription, but in another cell, it might be transcription factor A, transcription factor D, and transcription factor E that cause transcription. In other words, it could be that you always need transcription factor D, you always need E, but you could use either B or A. And it could be that if there's binding by TFC, that always represses. So the result could be, at one stage of development, TFB, TFD, and TFE as proteins, all present, you wind up with protein X. And at a different stage, at a different place, it might be A, D, and E, and the result of the combination is protein X. The result of this is it means that different combinations of transcription factors can mean that early in development, maybe you have transcription factors A, D, and E, and that causes protein X to be expressed. But later, maybe transcription factor A is no longer being expressed, but in some cells, transcription factor B is being expressed. Now in a different cell, doing something different, you can have protein X expressed. I've shown 5 transcription factors here, but with dozens or hundreds of transcription factors, it's easy to imagine how the same target gene, gene X, could wind up being expressed in many different cells, and in each category of cell, it's a different specific combination of transcription factors that causes transcription of gene X and translation of gene X.